Hey everyone, Mr. Murphy here. I'm going to go over 6.2, which is all about cellular respiration. I will have a separate video, which will be more specifically sort of how we should be thinking about cellular respiration, but I still think it's helpful to just run through sort of the textbook material to give you just a quick overview. So while this is a huge topic, it's not going to be that many slides, and I'm going to skip through a lot, but I do think there's some better visuals that we could use that we talk about in class. So I'll have a separate video that sort of does a deeper run through with that. Cellular respiration is the breakdown of our carbohydrates. And so it turns out there's a lot of energy released when a molecule that has a ton of carbon-carbon or carbon-hydrogen bonds is completely oxidized until it is just CO2. And that process involves three big steps: glycolysis, pyruvate oxidation, and the citric acid cycle. Now you may be thinking, wait a second, the electron transport chain is missing. And it is. But that's kind of a separate thing to the arc of the story that's going on here. And so don't worry, that is something we will discuss. The process of releasing the free energy of glucose is kind of interesting. In the mitochondria, we see the diagram on the left where it is many reactions that each have small little activation energies, enzymes to bring that down, in order to ultimately release that carbon dioxide. But a fun fact, and something you probably shouldn't try at home, you can take carbohydrates and you can just make them explode. And you can make this whole process happen very quickly with a larger amount of activation energy from something like fire. And you can look up videos on YouTube, just search like flour exploding. It's the reason why like old granaries and things in like even medieval times would literally explode and blow these things apart. Because when you get a carbohydrate in the right state, which is to say like a very small form, and have it thoroughly mixed with oxygen, like the dust of flour in the air, that's actually very explosive. Keep that in mind next time you're doing some extreme baking with open flames nearby, or just don't do that. So the process is shown here, where we have the process of glycolysis, pyruvate oxidation, and the citric acid cycle. Now, all of that is feeding then the process of electron transport, which is where the ATP is meaningfully synthesized. We make some ATP from these other parts. You may remember that from previous times you've gone through this. But ultimately, what's being produced is CO2 and water. Now, those are waste products. They're not the sort of intentional function of the mitochondria or the cell at large, but they obviously serve a purpose in the broader cycle involving photosynthesis as well. Glycolysis is 10 different reactions. It's a lot, and it takes place in the cytoplasm, so in the plasma of the cell. The final products are 2 pyruvates, which are 3 carbon molecules. That should make sense, right? We take a 6-carbon glucose and we turn it into 2 3-carbon pyruvates. It makes 2 ATP and it makes 2 NADH. Remember, that's our electron carrier that has energy potential, which gets utilized in the electron transport chain. And here you can see, this is where I'm going to kind of go through quick. It has activation energy. It requires 2 ATP, but we'll see that the 2 molecules that are produced, the 2 3-carbon molecules that are produced, actually release 2 ATP along the way, or allow the formation of 2 ATP along the way is a better way to say it. The process involves oxidation and reduction, and it involves ultimately this molecule being broken down, changing the placement of these different phosphate groups and releasing the potential energy of the bonds that they contain, ultimately yielding the results of glycolysis. So this is kind of the more specific chemistry that we don't need to pay as much attention to for where we're at right now, but it is important just to recognize that it is happening in this way. That pyruvate that's produced then needs to be oxidized, which is facilitated with something called coenzyme A, which you'll see written as just CoA, and that is how we get what's called acetyl-CoA. I bring this up just because a lot of times we sort of neglect it, or it's just sort of a step that's happening on the page, but we don't talk about it a lot. Glycolysis yields pyruvate, And then a lot of times we look at the Krebs cycle and we say it starts with acetyl-CoA. So we had to get from pyruvate to acetyl-CoA, and that process is where we produce some NADH as well. The citric acid cycle, also known as the Krebs cycle, is 8 additional reactions. For every glucose molecule that comes in, it operates twice. Should sound familiar. Calvin cycle operates twice for every glucose it's producing. And so it starts with acetyl-CoA, like I said, and we start breaking that down and start turning it into CO2. We start breaking the carbons off, and you notice if you look at the carbon count here, we have our 2 carbons coming in. I think I neglected to mention that a carbon dioxide comes off during the oxidation event here. So we have 2 carbons coming in. We have this 4-carbon molecule that's scooping up those 2 carbon molecules, becoming a 6-carbon molecule, and then stuff starts happening. We start breaking these things down, and in that process we produce NADH, we produce CO2, and then we also produce another NADH over here and an FADH2, which is another energy carrier, electron carrier, that will be used in the electron transport chain. The ultimate result here is that we're bringing in these carbon molecules, and we're liberating those individual carbons. So for every 2 carbon molecule that comes in, we release 2 carbons as CO2, and we make a bunch of energy carrying molecules. So we are the exergonic release of energy from the breakdown of this is being stored as our energy carriers. It also produces a little bit of GTP, which we kind of consider to be synonymous with ATP. It's not. That's biologically and chemically inappropriate to say, but kind of for our purposes, it's an energy carrier that fuels different reactions inside of the cell. There are different enzymes and kinases and things like that require GTP versus ATP. It's just a difference in sort of the base of the molecule and then what the 3 phosphates are actually attached to. So it's not a meaningful difference that we need to harp on for the purpose of this class. The final reaction is to produce this oxaloacetate. That's a word you don't need to know, but it's similar to the ribulose biphosphate that is scooping up the carbon dioxide in the Calvin cycle. So we similarly need to get like our acceptor molecule ready to do the next cycle. And so similar to the Calvin cycle, we kind of have a little bit of a regeneration here where we're putting everything back to how it started so that we can continue the cycle anew. Okay, then we get to the electron transport chain, and this is where we take NADH and we take the FADH2, and basically we are oxidizing these and releasing their energy, they're releasing their electrons, which are then passed along through a series of proteins, just like we saw with the light-dependent reactions of photosynthesis. And this is a process by which ultimately we're using the energy of these electrons to pump hydrogen. We're pumping hydrogen through the mitochondrial membrane, so between the inner and outer membrane of the mitochondria. Remember, mitochondria has multiple membrane layers, and so those electrons get dropped off, they bounce through some different proteins, and they use all of their potential energy to facilitate the movement of hydrogen ions. When that happens, now we have a lot of hydrogens here. ATP synthase steps in, and that's where we are doing some serious powerhousing. We are producing, through chemiosmosis, massive amounts of ATP. When we, so you see that, what I just said, about 32 molecules of ATP are produced for every fully oxidized glucose. Now that is, it says about, chemistry is not perfect, and the exchange between sort of, well, how many electrons are being dropped off and how many protons does that mean, and how many protons really need to pass through ATP synthase to facilitate the making of ATP. It doesn't all work in round numbers, and so that's why we have sort of like an average amount. You can see it referred to as 32 to 36 ATP or 32 to 38 ATP for like one full round, because we are making some ATP in glycolysis, and we're also making ATP or GTP in the citric acid cycle. So don't worry about having like a firm number. Just understand that it's in the 30s, and that's good enough for us. That is not photosynthesis. That is cellular respiration. It's a lot, but like I said, we'll look at some better diagrams that will help us to understand it better. If you have questions, let me know.